My name is Fraser Sinclair. I'm a researcher at the University of Edinburgh, and I'm going to be talking about some recent work we've been doing on modelling ecological links using phylogenies for gall-forming herbivores and their parasitoid natural enemies. All organisms exist within communities of interlinked species. And these links may be mutualistic, such as for pollination or seed dispersal, but perhaps most frequently they are antagonistic, such as for herbivory, predation, parasitism and parasitoidism. I'm particularly interested in the structure and evolution of ecological links between plants, their insect herbivores and their parasitoid natural enemies. Today I'm going to be talking about insect herbivores and their parasitoids, but many of the principles and methods apply equally to other types of ecological link. The ecological links between insect herbivores and their parasitoids are mediated by traits on both sides. These might be behavioural, such as when and where a parasitoid will search for its host, or they might be physical, such as hosts that induce or possess physical barriers to protect against attack. When considering the evolution of these traits across sets of species, it's essential to also consider the phylogenetic relationships of the species. This is because quite often we find situations like this where closely related species have similar or the same trait values due to their shared common ancestry and independent of any selection. In such a case, we would describe this trait as being phylogenetically conserved. Alternatively, where selection has taken place, there may be a situation such as this, where unrelated pairs of species have the most similar trait values. And in this case, we could describe it as phylogenetically convergent. Identifying and measuring the traits that mediate the interactions can be quite challenging, often requiring large samples of individuals and potentially being confounded by variation throughout the life cycle of an individual. On the other hand, estimating phylogenies is becoming increasingly straightforward, often possible by sequencing a handful of genes from just a single individual per species. Consequently, we became interested in this kind of situation. We've sampled a set of ecological interactions and estimated the phylogenies for species on both sides, but you don't yet know anything about the traits that are mediating the interactions. In such a circumstance, is it possible to build a phylogenetic model? And what does this model reveal about patterns of traits? Also, could such a model be useful for making predictions about unsampled species without actually measuring any of the traits? We set out to investigate this using a community of herbivorous cynipid gall wasps and their calcitoid parasitoid natural enemies. It's quite a good system for this type of question because the community is moderately species rich, but it's ecologically closed in that the gall wasps are specialist herbivores of oak trees and the parasitoids are specialist enemies of the gall wasps. Our data come from 6 sites in Hungary that were surveyed between 2000 and 2003 and include more than 27,000 parasitoid individuals of 54 different species, and these were reared from 60 different host species. Phylogenies were estimated from a single mitochondrial and a single nuclear gene, and the parasitoid phylogeny is shown here along the bottom axis, and the gall wasp phylogenies are shown here along the left axis. These gall wasps have 2 generations each year, a sexual generation that develops during the spring, and an asexual generation that develops during the autumn. The phenotypes of the same species can be quite different in the 2 generations, as is illustrated here for the species Neurotrus quercus picarum. This has a moderately sized spherical gall in the spring generation, and a much smaller, more numerous spangle-type gall in the asexual generation. Because of these differences, it's common to consider the 2 generations as distinct communities. In the heat map here, we can see the frequency of parasitoids reared from galls of the spring sexual generation here on the top and the asexual generation here on the bottom. This type of data can be considered in 2 formats. The first is the incidence of links, so the presence-absence of a link between a particular pair of species. The second format would be the frequency of links, so how frequently a link occurs between a pair of species. We did both and the results were much the same, and so here I'm just going to consider the incidence type data format. We developed a phylogenetic mix model based largely on this excellent paper by Jared Hadfield and others from 2014 in American Naturalist. This includes several terms to account for variation in the identity of links, The first of these is the cophylogenetic interaction, which captures the extent to which related parasitoids are tending to interact with similar sets of related host species. As illustrated by this diagram here, where we have 3 clades of related parasitoids along the top and 3 clades of related hosts along the bottom, and each parasitoid clade is interacting only with members of a single host clade. If we were to model this particular community, we would expect to then find a strong, clear cophylogenetic interaction term. We also fitted a parasitoid phylogenetic interaction term, which captures the extent to which related parasitoid species are interacting with similar sets of unrelated hosts. There's also a similar term that accounts for the same pattern but from a host perspective. And then finally, because our data incorporated multiple sites, we were able to fit a parasitoid host species interaction. And this accounts for the situation where parasitoid species are interacting with consistent sets of hosts across sites in a way that's not well explained by either phylogeny. This is an interesting term and we'll return to it in later slides. In addition to accounting for variation in link identity, Hadfield et al. Demonstrated that it's important to also account for variation in the number of species that a particular species will interact with. We refer to these as link richness terms, and the first of these is the parasitoid phylogenetic effect that captures the extent to which related parasitoid species are attacking similar numbers of host species regardless of the host phylogeny. So in this diagram we see that the parasitoid clade on the left is each member of this has 3 hosts. For the clade in the middle, each member has 2 hosts. For the clade on the right, each member has a single host. And if we were to model this, we would then expect to see a well-supported parasitoid phylogenetic effect. There's the same pattern, but from a host perspective. And then finally, there's 2 terms that account for any remaining variation in the richness of links between species. So the richness of hosts from a parasitoid perspective, or the richness of parasites from a host perspective. We fitted our models in the MCMCglmm R package and estimated the intraclass correlation coefficients for the various model terms fitted as random effects. We then inspected the median, the mode, and the 95% credible intervals for these, and used the cutoff threshold of 0.01 to distinguish between unsupported and supported model terms. For the spring generation gall dataset, the only well-supported model term, shown here in yellow, was the parasitoid host species interaction. If you remember, this is the situation where parasitoid species are attacking the same sets of hosts across sites, but in a way that's not well explained by either phylogeny. In terms of the traits that might be mediating these interactions, this is not what we would expect if the traits were phylogenetically conserved in either trophic level. The implication is then that they are divergent or convergently patterned. For the autumn generation dataset, again we saw a well-supported parasitoid host species interaction term, but the strongest term was the cophylogenetic interaction. And this is the situation where related parasitoid species are attacking sets of related hosts. Now this is, in terms of traits, this is exactly what we would expect if the traits that were mediating the ecological links were phylogenetically conserved in both trophic levels. Now in terms of predictions, it might be useful to use this type of model to predict either the parasitoids that are associated with an unsampled host, such as if we had an invading host and wanted to predict its natural enemies, or if we were to run a biological control experiment and wanted to release a parasitoid, could we perhaps predict which hosts it might target? For the phylogenetic models to be useful for this, it's important for them to have a strong phylogenetic component in the estimation of link identity. So these 3 terms here at the bottom. For the spring generation model, the lower credible intervals for these terms was all low, and therefore we would expect the predictive potential for this model to be quite low. Comparatively, for the autumn generation dataset, this strong cophylogenetic interaction means that there could be some predictive potential for the model of this dataset. Another useful aspect of the MCMCglmm output is that it estimates an effect size for each level of each random effect. And we can then use these to see which pairs of species are contributing towards the various effects. What we're looking at here is a heat map for the cophylogenetic interaction term of the asexual generation dataset. This is the interesting term that we highlighted on the previous slide. And in this case, strong positive values shown in red represent pairwise interactions whose presence is contributing strongly towards this term. And a low negative, well, a highly negative value shown in blue would be where the absence of such an interaction is contributing towards this term. To pull out a particular example, this red spot highlighted here indicates that ecological links between members of the parasitoid family Pteromidae and hosts from this clade here that includes the genera Trigonaspis and Cynips. These positive associations are contributing towards the cophylogenetic interaction. But similarly, this area of blue squares here indicates that the Pteromidae are not associated with this large clade of gall wasps from the genus Androcus, and this is also contributing towards the cophylogenetic interaction term. In conclusion, our phylogenetic mixed models are suggesting that the traits that mediate the ecological links are weakly phylogenetically conserved in the spring generation gall community, but more strongly so in both trophic levels of the autumn generation community. In terms of the potential to predict ecological links, this is low for the spring generation gall community, but moderate for the autumn generation. This work is in the final stages of preparation for publication, and we expect to have a preprint available early in the new year. So if anybody's interested in the methods, then please get in touch via Twitter or email. For the next stages, I'd like to test the predictive power of the models using a leave one out cross-validation approach, and then also to start incorporating traits, particularly from the galls, to see if and how these can improve the predictive power of the models. This work is being conducted as part of a large NERC-funded project called TRICOM, which will eventually draw together data on plants, galls, and parasitoid natural enemies from Hungary, the USA, and China. I'd like to say thank you to our various collaborators, and thank you to all for listening.